Hello, so my name is Alžbeta Rástocká. Firstly, I would like to clarify there might have been a misunderstanding in filling out the forms. I am from Slovakia, but actually Palacký University is in Czech, in Olomouc. So I would like to shortly introduce you to our work on soybean genotype phenotype analysis in soybean. And before I get into that, let me start with why do we study soybean? So soybean among major crops stands out with its nutritional profile. It provides both high quality protein and oil content. And this makes it very important for food and feed production. The global demand for soybean continues to grow, and this puts breeders into challenging position where they need to develop more resilient varieties that combine good agricultural traits. However, the mutation discovery, the effective breeding stands on knowing which mutations are actually responsible for the traits we care about. And mutations are binary. The mutation in genome is either present or absent. However, many traits are not. For example, quantitative traits like protein and oil content, which you can see in these graphs, exist on a continuous scale. Another very important category, quantitative trait for soybean is maturity group, which reflects the time when soybean plants reach maturity state. And this quantitative trait can be divided into several categories. First one is category 000, which reflects soybeans that are the earliest maturing plants, and higher categories correspond to plants that tend to wait longer. They tend to wait for shorter days until they flower and reach maturity. And these categorical and quantitative traits often arise from variant positions in multiple genes, which makes it difficult to discover causal mutations. And this is where AccuRate, a tool developed for post-GWAS analysis, comes to play. It divides non-binary phenotypic traits into binary classes and then assigns each binarization split and accuracy score. This score or parameter reflects how well a mutation's presence or absence matches the binarized phenotype classes. The tool AccuRate was first validated on several genes which are already known to affect soybean important traits like I previously mentioned. And then it was applied to two new genes that were identified by GWAS analysis to be associated with soybean maturity time. While the mutation in first gene with its low accuracy values among all found splits did not show any correlation with this trait, the second gene stood out. One specific mutation in the second gene, an adenine to guanine mutation, shows strong correlation with actually later flowering and later maturing plants. All found splits showed over 80% accuracy, which provides a strong evidence on potential causal relationship. And identifying such mutations is important for breeders because they can select earlier or delayed maturity in soybean, which helps to improve soybean adaptability to local and various growing regions. And this brings me to the end of my flash talk. If you are interested in further details, please join me at my poster later. Thank you.
Thank you very much.